All right, so you all have been here for a couple hours and you've already gotten assigned homework. So that's great. So my body is I'm from East Coast US time right now, and so it's like 11:45 p.m. And then I've been here for a day, and so it's like lunchtime also. So, so there's a lot going on. So why don't we all stand up real quick? Yeah? All right, so just to get to know you guys a little bit better, could the physical oceanographers please sit down? All right, so we got two physical oceanographers. Chemical oceanographers? What's that? Okay, so a couple of those. Economists. One, two, okay. Ecologists. A couple ecologists. A lot of ecologists. I like that, all right. Plankton biologists. Nope. Oh, couple. What am I missing? Geological oceanographers, all right. So we got a couple of those. Sociologists. Yeah. And then, so what else am I missing here? I know I'm missing some big ones. Paleo, okay, paleo. Computer scientists? Computer science, all right. And then the lone person? Fish biologist, okay, great. So I feel, statistics, okay, another thing near and dear to my heart. So I feel like I know you all so much better now. So hopefully this isn't too broad, and hopefully it isn't too specific. I want to have an exchange here, so if you guys think that you have something that you can add, please go ahead and speak up or raise your hand, and we can include your feedback, because really I hope to learn from you all, and hopefully you'll learn a little bit of something from me. So here's the outline of what I'm going to be discussing. I guess I didn't say AV guys in the back, so I guess you can stand. So I'm going to talk about chemical and physical climate change. It's going to be a little bit of an overview, more broad than maybe what Beth discussed earlier, and then discuss a couple of case studies of how these changes have and might influence marine ecosystems, some data considerations that we might want to think about, and how we measure some of this change. So I don't know how well you can see this graphic here. This is from one of Scott Doney's papers. The highlights that you should notice, the red line here in the bottom is increasing. This is CO2, atmospheric CO2 level. And so the direct effects of increasing atmospheric CO2 is manifest in increasing acidity. So the blue line here, we see an increase in ocean acidification, and then an increase in temperature, which is this brown line that's going there. And so from these direct effects, we also have a couple of indirect effects, such as decreasing in levels of sea ice, increased ocean heat retention, increased sea level, and increased stratification. So these are all the direct and indirect effects that we can see and measure using physical and chemical properties. And so as we extend the time series here, we can see that the acidification is going to further increase and that the CO2 is going to further increase. And so as Beth mentioned, that these problems are probably going to get even worse. So here's a figure that was from the 2007 IPCC report. And this sort of shows that, so here's the North Pole, and here's the South Pole, and here's the equator. And so these changes aren't going to be the same across the entire ocean. They're going to be manifest differently in different areas. So reduced sea ice extent, and then we have changes in some of the large oceanic current processes. And so we can see that these changes are going to be different throughout. So when we have these different physical and chemical changes, these will go ahead and also be present, and they will affect the physiological functioning of these different groups. So fish might behave differently, as Beth mentioned, and so from these physiological differences, we're also going to see population and community level changes. So I'm going to discuss a couple of examples of how we can measure this. So here's an example. I tried to make this a little bit clear. So I'll walk you through the slides here. So these are all of the different direct and indirect effects. And so on my computer, this shows up as brown, this background, and so that has to do with temperature. So just to sort of clarify, so the main climate effect here is temperature. And so specifically in this paper by Malin Pinsky, that it's discussing the climate velocity. And so that's the rate and direction that climate shifts. So they measured an isotherm, and over time they measured how it shifted. And they also looked at the different communities in these different ecosystems. And so each of the vectors here, you can't really see the vector. Each of these little green dots or purple dots or whatever, that's a vector. And so that's a direction that a species shifted over the time series. So then for each of these ecosystems, we have the mean shift. So for example, down here in the Gulf of Mexico, I've already met one person who's spent some time in the Gulf, which is great. That's where I spent a couple of years. But so it's a westward shift, or we have a southward shift in the velocity of these communities in response to shifting climate. So we have these community level changes. Here's another example of increased stratification or changes in the stratification properties. So Jeff Polovina and his colleagues, they looked at some remote sensing data, and they've run a couple of models. So here's what the Pacific looked like between 1995 and projected into 2017. And so there's this black isotherm here that basically is very low productive water. And so there's Hawaii right there. We're somewhere over here. So it's a big isotherm that represents very unproductive water. And so through their simulations, they found that this increases by about 30%, whereas other more productive areas decreased by a similar amount. And so this signifies that there's going to be some real changes in different areas based on different things. So here's stratification. So we see an increase in some ecosystems and a decrease in some ecosystems. And so there's going to be winners and losers throughout. So here's another example of how we might expect to see some changes. This is the Gulf of, so this is Alaska. These are the Aleutian Islands right here. And the sea ice moderates the temperature. And so this is during more or less normal conditions, where you have this big tongue of cold water that is very persistent. And this separates a couple of demersal stocks. And so let's see if this works. So over time, this has decreased significantly. And so now this barrier that used to be present has disappeared. And so now we have communities that have changed. We've actually seen increased catches, which that's not bad, right? But we've also seen these drastic changes. This is the Arctic catch per unit effort. Oh, that helps a lot, thanks. The Arctic catch per unit effort over the subarctic catch per unit effort. And so we can see over this time scale that the changes are pretty dramatic. And so again, every ecosystem is a little bit different, but we have winners and we have losers. But generally, we have change, right? So another interesting example here is changes in ocean acidification. So this is another pretty interesting paper this is the California coast here. There's Oregon and Vancouver up there and down the California coast. I've either had too much coffee or not enough, so my hand's shaking. So the upwelling brings carbon-rich waters from the ocean interior into the shelf, and so this is pre-industrial estimations here. So this is the percent aragonite, or I don't remember exactly what they measured, but it can be, it's a measure of the anthropogenic CO2. or and so that's pre-industrial, and then this is what it looks like currently. So as we can see that there's a lot of areas that have much higher acidification, and so this is actually happening currently, and there's these little planktonic vores called and so here's low unsaturated water, and this is a nice healthy guy, and then this is one that has had a lot of shell dissolution. So as the percent saturation increases, so in the red areas, you're going to have a lot more dissolution, right? So yeah, see, that's what I need. I need some feedback here. So so we can see that there's going to be some major changes. Okay, so who cares about the pteropods? But we can look at this on a more broad scale. So this is a graphic of ocean acidification in 1850. So the lighter colors represent areas that have higher acidity, whereas the blue areas are more or less normal. And so over time, we can see that this isn't as fancy as Beth's animations, but this is all you get. So over time, you can see that the acidification has really increased. And so my colleague Gavin Fay, who's now a faculty at the University of Massachusetts in Dartmouth, he's done some Atlantis modeling, which Beth has developed. And so here is an ecosystem model according to different ocean acidification ranges. And so this is sort of a horrendous graphic, and he's since changed it. But basically what you can see is that these different ecological indicators and indicators of how healthy the fishery might be under different climate scenarios, under different acidity scenarios, they're vastly different. And so we're going to see, we could see huge changes according to some of this ecosystem modeling. So that, I think, is sent to Global Change Biology, so hopefully you guys can read that paper here soon. So to summarize, what we see are these physical and chemical changes, which are then transferred to physiological changes, which then are transferred to ecosystem changes. And so predicting from the temperature and all of these different physical conditions can be very difficult for understanding what the community changes are. But that's not to say that we can't do it, right? So does anybody have any questions about these altered structure, altered diversity, novel ecosystems? I think I've sort of touched base on each one of those potential community changes.
Scott,
yes?
Is there any possibility that these changes can also feed back?
Sure, yeah. You know, if we have changes in the ecosystem structure, that might change how the processes work. Exactly, yeah. Right. So there should be another arrow feeding back. Yeah. Yeah. So changes in the physiology. So if something is more or less metabolically active, that will certainly manifest itself down in altered diversity, because somebody's going to be, a prey item might be more vulnerable or less vulnerable, or as Beth mentioned earlier, the changes in ocean acidification can actually be manifest in the predator-prey interactions or the ability to find a mate. So all of these are highly interrelated, and yeah. So, yes? The community structure? Yeah. All of these are possible, but they're not necessarily going to be felt the same in each ecosystem. Some might be more pronounced in some ecosystems than others, yeah, and in some fish than others. So that's a good segue into the next bit. So what are the data limitations? What makes good data? So I started my professional interests off actually studying snails. So the predator-prey responsive snails. So that's exciting. So you know, when you sit down and start an experiment, what is good data? When you get out your notebook and you start thinking about how am I going to solve this problem? So it needs to be internally consistent. So that means that every observation of snail behavior that I watched had to be consistent, right? And so it also needed to span the range of natural variability. So for if there's a behavior that only happens at night, you should probably not only record data during the day, you should also record it at night. Internally consistent. So at the Northeast Fisheries Science Center, they have a fantastic bottom trawl survey that I'll talk to you a little bit more about later. But they do diet data. So they catch all of these fish in these big trawls and then they count them. And they rip out the stomachs and count what is in the stomach. And so these are hugely important data that are used in stock assessments and that are used in other things. But they can also be really important to gauge how communities are structured, who's eating who, these food web dynamics. And so the first half of the survey, they measured it, the diet components based on a volume, or on a weight, and then they changed that halfway through to make it dependent on a volume because that was more accurate. So there needs to be a way that you can move between one type of survey to another. So that's obviously they weren't thinking about that when they started the survey. They just wanted to get stomach samples, but it needs to be internally consistent or at least translatable between survey times or data collection times. And then it also needs to be have precision, and then it has to have so precision is there's accuracy and precision, right? And so you need to make sure that you have both, and you also need to have good variance. So that's high quality data. What makes useful data? So it needs to be directional, like what Alida mentioned earlier, all of the indices from the Indices Project, all of the indicators from the Indices Project. They're all designed that under negative conditions, they decrease, or under higher fishing pressure, they decrease. So it's important to have an idea of a directional response. It needs to be sensitive to change, represents phenomena of interest, and unambiguous. So so those are some data considerations. Does anybody have any comments or thoughts about maybe I missed some, or there are other considerations that might be important? I come from a ecological background, so I'm not really as versed in some of the other fields. So if there's other, for example, geological oceanographer, what would be some data considerations that you would need to focus on? Nothing. Nothing that you can think of. Okay. Yeah. Okay. So, all right. So anyway, so these are some data considerations, and so now we can go into a little bit about how some of these climate change indicators are measured. So this is a nice satellite view of the North Sea. This is continuous plankton recorder, which I'll discuss a little bit more. This is the number of observations for the continuous plankton observer mission. And then this is some ocean color data. So we have different types of climate signals that we can measure. One of the most widely celebrated, I guess, is probably remote sensing, where you have a satellite or a spacecraft or an aircraft that you measure the electromagnetic energy, and these are representative of different physical properties. And so then the frequency distribution of the received radiation is directly related to limiting conditions. So like what we mentioned earlier, temperature, sea ice extent, salinity, these are all things that can be directly measured. And then we can also measure things like primary production with chlorophyll A. So I'm not as up to date on the remote sensing, but some of the advantages are you have incredible spatial coverage anywhere a satellite can go, which is pretty much everywhere. But you're limited to the wavelengths that are that are available. Some satellites can, or some sensors are only sensitive to UV or different wavelengths of light. It has rapid temporal resolution for places that you couldn't otherwise get frequent measurements from. And then it also has really big picture, right? So we have the entire world that we can more or less look at the ocean color of. And then it's also easily compatible with GIS. You know, you can ground truth things. But the disadvantage is you have temporal coverage. It only starts in the early 1990s, mid 1990s. So we really don't have an idea of what was happening before that. Also, it's limited on the flyover, so how often the satellites fly over. Some of them, it's only 18 days, or it could be longer. You guys could probably comment more on that. And then it also requires a clear sky for some sensors. And so a lot of the ocean color data, for example, is pretty spotty up in the northern and southern latitudes. And it also requires validation. And then it also, there's problems with multiple explanations for a single change in ocean color. Is it a change in the sensor? Is it a change in the atmospheric conditions or otherwise? And then also there's interesting depth limitations. For the visible wavelengths, it's the depth average for the top 10 to 100 meters, whereas the infrared satellites just pick up the very, very surface. So you have to be sure to understand the limitations for the different types of measures. Is there any other limitations that people might think of for the remote sensing? How many people daily use remote sensing data? Weekly or frequently use? Yeah. Okay, so quite a few people. Alida? Yeah. Mm-hmm. Right, right. Right, right. You don't just open up the satellite file. Right, there's a lot of steps that are...
Yeah, what I've been told by biological astronomers is that at least 5 years ago, it still was like an estimate of phytoplankton productivity, for example, would be like plus or minus 50%. It's still very ballpark-ish.
Yeah.
Whereas we used to be dealing with more accurate estimates.
Right.
So it's important to bear that in mind when we're using that kind of data.
Right, and that goes back to some of the data limitations in general, is understanding what kind of precision you're able to estimate, and if that is within the range that you can actually use statistically.
Issues with respect to resolution.
Pardon?
Resolution of the image.
Right, resolution of the image, so how
Sometimes it is very broad, so coarse images we are getting. You don't have fine resolution.
yeah,
That will be disadvantage instead of
Yeah.
specific. Like,
no, that's a good point. Okay, so another type of measurement is in situ, so that's where there's a ship that goes out and samples something. And so one of the examples of this is the extended reconstructed sea surface temperature. One of the great things about this is that it's a gridded product that goes back to the 1950s and then more accurate up until the 1850s and more accurate into the 1880s. But this, there's certain limitations with this, obviously, because it, so you take your different known measures and then you sort of extrapolate over these really large areas. So what you lose in temporal resolution, you gain in having a really nice long-term data set that you might be able to use to make more general assumptions or use differently. Another in situ measure is the continuous plankton recorder. Has anybody used CPR data? A couple? Yeah? Alida has. So for those of you that don't know, it's a really ingenious little contraption that, how many years ago is it? It was like in the early 1900s that it was invented. But it's basically a little towed piece of gear, and it has a piece of silk that sandwiches plankton together, and then it fixes it in formalin. And so it's really, really simple piece of gear. And so that image that I showed earlier, that is the number of, So this is from 94 to 98. These are the number of observations that the CPR has recorded. So obviously you have interesting spatial and more or less temporal resolution issues here where, you know, if you're trying to estimate what's going on right there, you're probably going to miss some signals, or even in the Southern Ocean, you're going to miss some signals. But by and large, it's a really important piece of gear that has generated some really nice patterns. This is looking at harmful algal blooms and changes in harmful algal blooms over time. So that's another piece of gear that is really important as an indicator. So another way that ecological and fisheries indicators are through fishery independent surveys. And so this is the Henry B. Bigelow boat, and it goes twice a year up and down the northeast coast and samples all over the place. And so we have a really nice data set of size, production, diversity. And then we also have some canary type species, which are the canary in the coal mine. I don't know if that's something that translates across multiple cultures or not, but basically it's a species that is sensitive, and so we might expect a decline in that species or an increase in that species ahead of other less sensitive species. So as Alida mentioned earlier, so these are ecological indicators here that were proposed for the indices project. And so we have indicators based on size, trophodynamics, species-based interactions, pressure indicators, and biomass-related indicators. And all of these came from a type of fishery-independent survey. And so they can be really important in understanding all of those different aspects of a community. So obviously this is more fishery-oriented to look at the community responses to fisheries, but these are some examples of how some of those indicators are measured. Another way is to use fishery-dependent data. So this is another paper here by Jeff Polavina, and basically they used, so they're situated out in Hawaii, they're situated out in Hawaii where they don't really have the ability to do a very specific statistical sample survey. So they have to use their fishery log data. So they take the logs from the fishermen who do a pretty good job of reporting all of the fish that they catch. And so they use catch per unit effort estimates. And so here with this, you could actually notice that these are striped marlin, spearfish, shark, albacore, and big-eye tuna. So these are upper trophic level fish that are really highly sought after commercially. And here we have some mid-trophic level fish, long-nosed lancet fish, mahi-mahi, sickle, pomfret, escolar, and snake mackerel. And those aren't really as commercially sought after. But over this time series, they noticed huge increases in the catch. So they weren't necessarily retained species, but they caught these species. And so they're able to say, from these fisheries dependent data, we can see changes in the community structure. So this can be a very important aspect of measuring fisheries response also. And so in summary, we have these different chemical and physical conditions that change the physiology and the biology of the different organisms, and that is translated into different communities. And so I just briefly covered a couple of different ways to measure those types of community change or change in physiological adaptations a little bit. So it looks like there's still plenty of time for questions or discussion if anybody has any comments. Crickets? No?